Welcome everybody to the first masterclass of the new year. It's glad to have you all back. I hope you all had a really nice break. I'm really pleased to introduce today Eileen Gentleman. She's a reader in bioengineering here at King's. Just a quick academic background. She's got a PhD in bioengineering, 2005 from Tulane. She then did a postdoc at Imperial College London before moving to King's in 2011 to establish her own independent research group. She's been very successful here. Her research focuses on developing biomaterials for tissue engineering and disease modeling, and she does lots of really cool things that she's going to tell you about today. And in case Eileen looks familiar to you, yes, she was on the interview panel for the Mechanics of Life when you all interviewed, so you've all met her before. And so, so she's going to give you a lecture today, and I really encourage you to participate, ask questions. I'm sure Eileen wouldn't mind you interrupting. There's no question too big or too small. This is really, for you to, learn as much as you can. So I hope you enjoy. I'm going to duck out, but I'm going to pass over to Eileen. Eileen, thank you so much for being here today.
Okay, so I'm really happy to be here today to tell you about, I guess, my field, and what I'm going to try to do is introduce you to my field. So some of you might be quite familiar with some of the background I'm going to give you and some of the research methods, but for others it might just be new and interesting. And then I'm going to end by sort of telling you a bit about work we've done over the past few years to sort of contribute to this field. And I don't know where I came up with this title, but I guess it sort of covers a bit of what I'm going to talk about today. So I guess to begin with, I'll tell you where my research career begins, which is in this field of tissue engineering. Is everything okay? Because now I definitely can't see you guys. If anyone's turned on their camera and is waving, I don't see it. So it's in this field of tissue engineering, which is this idea that we could grow tissues in vitro to try to replace those that have been damaged or lost due to disease. And the basic idea of how people have always thought you could do tissue engineering would be to take cells from a patient, expand them in the lab, and then put them in some sort of 3D structure, normally called a scaffold, with some sort of cues that would then make the cells, and people would normally think these are stem cells, differentiate and form a tissue, and then you would culture this and put it back in the body. So this has always been the idea, and this was the idea that was sort of a new field sort of when I started my PhD, and the whole idea was introduced probably about 30 years ago. And so that of course begs the question, what in many ways are these little squiggly lines? What are these cues you would need to make this happen? And so obviously we know from biology that there's loads of cues that we know, biological cues that cells receive that we know impact their behavior. And specifically we know this, or I don't know if we know it really well, but we're learning more and more about how this works with progenitor or stem cells to create the adult organism. And we know there's loads of biological signals, growth factors and such that drive that. But in terms of other cues that impact stem cells and the response they have, their differentiation, you can think, as I said, there's all these things like secreted factors, growth factors, but there's all cell-cell communication. There's also things like hypoxia, metabolism, inflammation, etc. But from the point of view of the tissue engineers who are trying to build a 3D tissue, what we have are these factors down here. You can try to mimic the extracellular matrix. This is the proteins and sugars that surround all of our cells, and try to manipulate this because we know these cues have an impact on stem cells. Or you could try to mimic physical factors of the cell's local environment using material science approaches, because we know, and I will talk about how some of these other physical factors themselves influence stem cell differentiation. And this one here has been probably the most exciting for tissue engineers, because if you're creating a material, you essentially don't have many biological cues, but you can change these physical cues quite easily. But in the context of all of that, we do have to remember that most cells, when they're in the body, most cell types are within 3D matrix. And so here in this image, it's showing you what looks like 2D. And of course, a lot of this work is in 2D, but that actually doesn't replicate what most cells experience in the body. So this is, I'm sort of showing you a summary of intrinsic mechanical cues and how they impact stem cells before I actually show you the data on this. And I show you this figure because I just want to make clear what I mean by intrinsic versus extrinsic mechanical cues. So we know, going back to the 1860s, that extrinsic cues like loading, actually physically applying loads to cells, impacted the cell response. And that was documented by Julius Wolff, who was a physician in Germany, as I said, I think like 1860s. And he had reported that in patients who had displaced hip fractures, so essentially they broke their hip and then it was not surgically fixed, because obviously that wasn't an easy thing to do, more than 100 years ago, the displaced fracture would reheal and you would get changes in the orientation of the matrix that forms the bone in these healed fractures over time. And that's an effect of extrinsic cues, in this case, mechanical loading, impacting cells and how they create matrix. And this is also a big field, for example, in endothelial biology, so the cells that line blood vessels, how fluid flow through the vessel itself impacts their response. But what we're interested here, and what I'm going to talk most about, is these intrinsic cues. So what I mean by this is things like the shape of an individual cell, the stiffness of a matrix that a cell sits on, and I'll talk less about this topography, but if you create a topography on a surface, these all influence a cell, and it's and in many ways a stem cell, and these are the evidence I'm going to show you and how it differentiates. And the way this works is summarized very simply here. It's obviously far more complicated, but the basic context of it is you have mechanical cues. These, in some way, most of them converge to alter cytoskeletal tension, which directly affects the nucleus and transcription within the nucleus, and that this then drives some sort of cellular behavior. Before I go and I show you some of the first evidence of intrinsic cues and how they impact stem cells, I just want to introduce the type of cell that many of these studies has been done on, early studies. And I mean early, like 15 years ago, because this is not a very old field, but they were mostly done using human marrow stromal cells, what are sometimes called mesenchymal stem cells. So these are basically fibroblastic-like cells that exist in the adult bone marrow, and they're not well defined. If you are in this field at all, there's a lot of criticism that they're not well defined like other stem cells that I'll talk about later, but these are very much used in many of these studies. They have the capability, people have reported, to differentiate into osteoblasts, adipocytes, and also perhaps chondrocytes. And what I'll just tell you before we get into here is there's long been known that progenitor cells sort of can make a fate switch between being an osteoblast versus being an adipocyte, a fat cell, and that there's a few transcription factors regulating that. So people think that these two are sort of cells choose one fate or another, and that's sort of the basis that a lot of people used for many of these early studies. So the first one I'll show you was a paper that was published just in 2004 in Developmental Cell from a lab in, they're now in Boston. I believe they were at UPenn at the time. And what they did is they took these MSC, these human bone marrow derived mesenchymal stem cells, and what they did is they used a technique called microcontact printing. And so what this does is you take a 2D surface and you make it mostly non-adhesive, so a cell won't stick to it. And then you make little islands that a cell will stick to. And if you do that and you make a very small island versus a very large island, when you put these MSC on this surface, they'll essentially adopt the size of the island. They'll spread out to either be big or small. And when they did this in the presence of chemical factors that would allow for both the differentiation of osteoblasts and adipocytes from these MSC, they found that essentially the size of the island itself would determine the differentiation. So cells that went on large islands almost always became osteoblasts. That's what's being shown over here. Whereas cells that went on the small islands almost always became adipocytes. And they were able to show that this was driven by the cell's ability to generate cytoskeletal tension through the spread cytoskeleton, which impacted the Rowe-Rock pathway, and that this was driving this differentiation phenotype. And so things like this were really exciting for people in my field, because here was, we had this whole field of biology talking about how it's this growth factor and this growth factor and this soluble signal, which is going to control a cell behavior. And here was an effect driven by a pure physical property of the material that was being presented to the cells. The next area that people really looked at after this was looking at the impact of substrate stiffness. So stiffness, again, you guys have a lot of you have mechanics background, so this isn't really new to you. Stiffness is, of course, exactly what you think it is. It's the resistance of a material to a deformation. And if you normalize the size of the material and the deformation, you can calculate something called modulus. In this case, for certain materials, we can call this elastic modulus or Young's modulus, which is basically a size-independent measure of the stiffness of a material. What is the intrinsic resistance of a material to deformation independent of its size? So you know that like a paper clip has less stiffness than a large block of steel, but intrinsically those materials have the same material properties. And if you can calculate the modulus, they will be identical for the two materials. So you can imagine lots of tissues in the body have different stiffnesses. You can imagine something like fat would be very soft, so it doesn't take much stress, as in you don't have to apply that much force to it for it to have a lot of strain, for it to deform that much. Cartilage would be stiffer and bone stiffer still. And people have made measurements like this on tissues. And as you can imagine, things like brain tissue and lung tissue are quite soft, in the range of hundreds of pascals. This is the unit of elastic modulus. Whereas you can imagine tissues start to get stiffer as you get up to smooth muscle and skeletal muscle, cartilage and bone. These all have increasing stiffnesses. And so what was noticed, again, about the same time as this last paper I showed you, was that there was this idea that cells themselves could be responding to stiffnesses that were similar to the native tissue that you would expect them to reside in. And this was really shown early on in skeletal myoblasts. And the way they did the first experiments is a bit funny to think of now, but it was done like this. So what they did is they cultured a layer of cells on glass. So glass is obviously very stiff. They cultured a layer of cells on glass, and then they cultured another cell on top of it. Now, the reason why they were looking at this is because it's quite well known that skeletal muscle cells, if you put them in culture, you put them on tissue culture plastic and you try to grow them in vitro, they'll lose their phenotype. So they'll lose the standard markers you would expect them to express. They sort of become a cell that's no longer what it was when it was in vivo. And so they were hypothesizing that this had something to do with the stiffness of the tissue culture plastic that you would normally plate these cells on. And so what they did is they plated the cells on the glass or the tissue culture plastic, and then they plated another layer of cells on top. And then they looked at basically an immuno for myosin, which would be a marker of a mature skeletal myoblast. And what they found is if they looked at the lower cell, so the one that was directly adhered to the glass or plastic or whatever this is, it lost its expression, as you would expect, of the myosin, but the one on the top didn't. It maintains its expression, and this is it, these striations quantified here, that the upper myotube had this. But of course, this is sort of an ugly way to do this sort of experiment. It's really exciting because it's suggesting that somehow the upper cell, although the soluble conditions are identical, so the medium, they're all experienced, the nutrients, the growth factors are all identical, the upper cell is feeling the lower cell, sorry, whereas the lower cell is feeling glass or plastic underneath. And so this is suggesting that the ability to feel a cell, which they claim was much softer in this range, 12 to 15 kilopascals, was impacting this one's phenotype. But as I said, it's very difficult to do this by culturing cell layers on top of one another. And so the field has come up with ways to create surfaces to do this more precisely and more cleanly, so that you don't have a lower cell, for example, that you wouldn't necessarily be able to modulate its stiffness. And there could be other biological effects, of course, going on from doing a culture system like that. And the way you make these materials now with defined stiffnesses is by using something that I'm sure you all see in the lab every day, which is polyacrylamide. So polyacrylamide is a polymer that you can polymerize, and people use it to do things like run electrophoresis, like this, which you can see here. But you can also essentially do a fairly simple protocol to stick polyacrylamide onto the surface of a cover slip in a thin layer, and you can then modify the amount of the base of this material and the crosslinker to change the stiffness of this thin layer that then gets attached to a cover slip. And then you can use also a rather simple chemistry that allows you to covalently crosslink an extracellular matrix protein onto the polyacrylamide, because normally a cell would never adhere to a polyacrylamide because you need some sort of matrix protein or something like that the integrins in the cell can recognize to adhere onto it. And if you do this, you essentially now have cover slips with defined stiffnesses, because you can of course measure these and know what their stiffnesses are, that are thin and optically transparent, that you can use for cell culture to start to explore what impact stiffness itself has on some sort of cell response. And in this case, again, we're going to look at the human mesenchymal stem cells. So people had known, I guess, since the sort of the late 1990s, that if you put a cell on a stiff substrate, it behaved differently, just visually, than one did on a soft substrate. Basically, what you got is if you put cells on stiff substrates, you got larger and more stable focal adhesions. So you can see here, this is glass, this is a stiff gel, and this is a soft gel. So on glass, cells will form very pronounced focal adhesions. You will also get them maybe not quite as much on a stiff gel. It's still much, much softer than glass. And then you basically, it's hard to identify focal adhesions if you have cells on very soft gels. And the actin cytoskeleton does a similar pattern. You see it's very defined. Stress fibers form when cells are plated on glass surfaces. They're very defined again on stiff hydrogels, and you have a more difficult time identifying an F actin cytoskeleton on very soft surfaces. And so with this, there was a group at UPenn who went and said, right, so why don't we look at the impact on MSC differentiation of plating cells on surfaces that match the stiffness of the native tissue, and let's see what they then become. So what they did is they created surfaces that were soft, 0.1 to 1 kilopascal, which is similar, for example, to the stiffness of brain tissue. They created intermediate stiffness polyacrylamide gels on cover slips, similar in stiffness to what's been reported for skeletal muscle. And then they made stiffer ones. Now here they claim that this stiffness is similar to that of the developing osteon. So mineralized bone has a very, very high elastic modulus, megapascals to gigapascals. But the way bone forms is you have individual units called osteons, which are essentially a collagen matrix that then gets mineralized by the hydroxyapatite. So what they were proposing here was that this was the approximate stiffness of the developing osteon. So, and they call that the bone stiffness. What they did, and you'll all forgive sort of the old school gene array here, because this was really before we had RNA sequencing as a widely available technique. What they did is they cultured the cells under identical soluble conditions. So the identical concentrations of growth factors in the medium. I think it was actually growth factor-free medium, if I remember right, but identical soluble conditions. And then they used these chips that were for either this one here, neuronal differentiation, this one here, skeletal muscle, and this one here, bone. And what they have are a list of genes here that one would expect to be upregulated if you had neuronal differentiation. And what they found was on these soft surfaces, they had red here is upregulated. Lots of these genes they would expect for neuronal differentiation were upregulated. When they looked at this intermediate stiffness, and here's all of the skeletal muscle genes, you can see a few of them here, PAX7 and such, they were the ones that were upregulated. And when you looked at bone-related genes, so this one here, this is osteopontin, that's a protein that's specific to bone. CBFA1 is RUNX2, it's a transcription factor associated with osteogenesis, and so on. These were all upregulated on the stiffest matrix. And in all cases, if you treated the cells with blebistatin, which inhibits non-muscle myosins, so the cell can't generate tension through its cytoskeleton, you completely lose this differentiation effect. So it was really exciting that basically you were differentiating cells, not using soluble cues, but rather these matrix cues. And they were able to show this again using some immunocytochemistry, that they had preferential staining for this marker beta-3 tubulin in the cells on the softest matrix, MyoD, which is a marker for the skeletal muscle here on the intermediate, and CBFA1, this is a transcription factor associated with osteogenesis here on the stiffest matrices. And then they could put this all together and they were seeing this trend. And the important thing that they pointed out as well was that this differentiation was enhanced on substrates that were mimicking those of the native tissue, suggesting that the differentiation and the native tissue stiffness were sort of going hand in hand. And a number of years later, people sort of figured out how this was happening, and at least one of the pathways that was governing this. And this was the Hippo Pathway. So the Hippo Pathway had been discovered by developmental biologists long before as a regulator of organ size. So this is just an example when you knocked out one of these components of the Hippo Pathway, you would have in mouse models, the liver would massively overgrow in size or something like that. But it was found that when you were culturing cells on either these stiff versus soft surfaces, or you were culturing them on these small versus these large islands, the differentiation was being regulated by these two co-transcriptional regulators called YAP and TAZ, which are essentially turned on or off in the Hippo pathway. And on or off means they go either into the nucleus, where then they're active and they drive transcription and will drive, for example, osteogenesis, or they're proteasomally degraded and then they don't go into the nucleus, and then you get, for example, adipogenesis. And so this figure here sort of sums all of this up. So if you have cells, and this is definitely true for MSC, and people have looked at this in other cells as well, growing either at high density or on a small area, or you put them on a substrate that's soft, a soft ECM, and the cytoskeletal tension is low. You essentially have cytoplasmic yapentase, so it's degraded, and then you get the cells undergoing adipogenesis. If you have high stiffness, so very stiff ECM, so the cells generate a lot of cytoskeletal tension, or you grow them on a large area or at low density, so they spread and generate tension through their cytoskeleton, then the yapentase enter the nucleus and you get osteoblastic differentiation, among other effects. So this was all really exciting for the field over the past years, but going back to what I said in the very beginning of how do we try to use this for tissue engineering, we have to remember that all of these studies I've shown you so far have all been done on 2D, but actually most cells reside in 3D. And so how do we translate this to 3D? How do we understand how to use this, or whether it really has this sort of impact we're seeing in these 2D studies when the reality is cells actually exist in 3D? And the answer to try to do this for many labs, including my own, is to use materials called hydrogels. So hydrogels are exactly what I've written here. They're water-swollen polymer networks. And so they're, it can be made from natural polymers, things like collagens, but also from synthetic polymers, things like polyethylene glycol. And they're like jellies, just like the thing you would eat, but you can encapsulate cells inside them so that cells can feel that network in 3 directions. And I'll tell you about how we can modulate them for experiments in exploring things like the impact of mechanical cues on stem cells. So the first study I'm going to tell you about used a type of hydrogel derived from a material or formed from a material called alginate. So alginate is derived from a seaweed, and the way you make a gel out of it that you can then put cells inside to sort of ask these questions is the polymer itself has these repeating blocks of M and G blocks, and if you place these chains in the presence of calcium ions, they'll assemble through sort of an ionic cross-linking process and make a network that you can then use to encapsulate cells. And by changing the concentration of these calcium ions, you can change the stiffness of these hydrogels. Cells themselves can't recognize alginate. It's a seaweed, and so it doesn't have integrin binding sites where cells can attach to it. And so in order to make it usable for cell encapsulation so that cells don't just die of anoikis. So cells can die if they're supposed to be adherent and there's nothing to adhere to, they can die in a process called anoikis. You put peptides on the alginate chains that have the RGD sequence. The RGD sequence mimics that that cells adhere to when they adhere to fibronectin. So it's mimicking the binding sequence in fibronectin. And then they took these alginates and then they made them of different stiffnesses. So down here, you can see they go from 2.5 kilopascals, relatively soft, up to more than 100 kilopascals. And then they encapsulated these the human mesenchymal stem cells inside and actually found what was found with 2D stiffness, which is that in the softest gels, you had adipogenesis, so which is what we had sort of expected from before. Fat is soft, so HMSC differentiation would be towards adipogenesis in soft hydrogels, and then in stiffer hydrogels, they were getting osteogenesis, cells becoming osteoblasts. So it correlated with what was seen before in 2D, but it was different than what was seen when people had grown MSC on small and large islands, because in that case, cells changing their shape and being able to generate cytoskeletal tension was driving differentiation. In this case, with these alginate hydrogels, cells always stayed round, but they were still differentiating in response to stiffness. It turns out that wasn't the whole story, and that's probably specific to ionically cross-linked hydrogels, because a few years later, using a different hydrogel, which was formed from hyaluronic acid and cross-linked using a covalent cross-link, another group was able to show that if they modulated the stiffness of these hydrogels, cells always became adipocytes. So basically, cells were insensitive to changes in stiffness, but if they allowed degradability into the hydrogel by introducing a secondary chemistry, then cells could spread and generate cytoskeletal tension, and that this was driving differentiation. So I guess what you could say about the 3D is it basically became far more complicated than what we see in 2D, which was probably also expected, because 2D is very reductionist and simple to think that that would replicate what we know in 3D to be very complex processes. Was probably something that could be expected. And there's also subtleties associated with the hydrogel chemistries here. But that, again, wasn't the whole story about how cells detect these cues in 3D. And this goes back to work that was done, almost 40 years ago by a researcher called Mina Basel. And she was studying the mouse mammary pad. And what she had described was something she at the time called dynamic reciprocity, which was this idea that cells in the body receive cues from their surroundings and then respond to those cues by changing their gene expression, creating matrix, modifying their surroundings, which then again signals back to them. So this idea that cells are in constant communication, receiving signals and modifying their surroundings and receiving signals. And the way that you can sort of describe what had been done so far with both the 2D surfaces and these hydrogels was the basic premise of all those experiments, were that cells were sort of passive sensors of their surroundings, that you could give them cues from a material, stiffness or shape or something like that, and they would just passively sense that. But from what Mina Basel had reported a long time ago, and things we knew in the biomaterials field about how cells rearrange proteins on their surface and such, there's now been more work suggesting that this idea of passive sensors is probably not true, and that cells are probably doing something that's more close to this dynamic reciprocity. And the way this was shown, this is some work that was done by my group, and others have shown it now as well, which is that if you put MSC in a 3D hydrogel, and what we've done here is used a relatively simple technique where you replace one of the amino acids in your cell culture medium with a non-canonical version of that amino acid that has a little functional tag on it. Then all the new protein that's synthesized by your cell now contains this amino acid, and then you can just use a very simple click reaction to identify it and put a fluorescent tag on, and then you can see it by microscopy. And what you find is very quickly after encapsulation, cells put loads of proteins around themselves, and how they distribute that protein and how far it goes from the cell is dependent on the hydrogel properties. But essentially, this whole idea of the passive sensor is probably, again, not true. And this was again, shown by other labs, and this is very beautiful pictures, that cells start to secrete matrix around themselves within 1 day, and within something like 6 days. They're not only secreting matrix around themselves, but modifying their surroundings quite dramatically. And that this, and it's been shown, although I don't show you that data here, this process itself has an impact on cells' differentiation. So going back to what we talked about with basically the first slide, that what can we do as a tissue engineer? We can try to mimic the extracellular matrix, cells receive signals, or we can try to create a biomaterial that replicates this. But the idea that the cell just receives this signal and has some sort of response is probably not necessarily completely true on its own. And instead, the cell sort of does this bidirectional interaction between the matrix you give them and their own secreted matrix and how that alters their surroundings. And together, this process is what determines the cell response. So now I'm going to just sort of spend the rest of my time with you sort of giving next generation examples of how all of this has been coming together over the past few years to allow us to create tissues in vitro, to actually discover something about biology, to discover something about disease. And I'm going to give you an example that was published a couple of years ago, and then an example from my lab as well. And both of these examples I'm going to give you focus on using a tool called an intestinal organoid. So we've known for a long time the gut is obviously highly complex. It's covered in this very complex epithelium that is responsible for all the nutrient absorption from your food in your body. It also houses a massive microbiome. You hear all the time that there's more microbiome cells in your gut than there are cells in your entire body. But it's been very difficult to study the intestinal epithelium for a long time because you can't culture these cells long term in vitro. They lose these distinctive cell types that you have in the intestine, and so it's very difficult to study. The way the intestine works is you have what is called a crypt-villus structure. So I'm sure you've seen pictures of the intestine before. You have these little fingers that sort of stick up from the surface, and those little fingers, called villi, are covered in epithelial cells all along them, and that creates this large absorptive surface for the gut. And the cells on these villi are renewed basically about every 5 days by a stem cell pool that resides in a little crypt or pit at the bottom of the villus that has a stem cell population that's constantly self-renewing and creating new cells that push up along the villus and renew all the cells, and then the dead cells come off the top. So what was discovered in 2009 was that if you could isolate these stem cells from the crypt of the epithelial layer that covers the intestine, and if you put them then in a 3D matrix, they would proliferate as like a little sphere. And then over time, they would make these little buds with the stem cells at the base of these little crypts that would bud out. This is what one really looks like here, and you would have stem cells down here. And then they would do, just like in many ways what happens in the normal gut, you would have differentiated cells arise from these crypts and then form these villus-like domains. And this was brilliant for the field because it really allowed people to, for the first time in vitro, to study gut development, tissue homeostasis, disease, repair. It was massive excitement. You could do drug screening, lots of things with this. So the way you form these organoids, and I probably forgot to say, these things will essentially grow indefinitely. So you can keep propagating them in culture. There's two ways basically to form them. You can either form human or from an animal. You can basically take a biopsy from a human or the gut of an animal, usually a mouse. You could isolate these crypts that contain these stem cells, which are LGR5 positive. You put them in essentially a naturally derived hydrogel called Matrigel, which comes from a tumor that is grown in a mouse, and then they will form these organoids that contain epithelial cells. You can also do this by taking pluripotent stem cells, usually again human, that have been reprogrammed from an adult cell into these pluripotent cells, and then you give them a series of growth factors that mimic intestinal development, and within Matrigel again, and then they'll form organoids. These ones from pluripotent stem cells, they have the epithelium and they also have the mesenchyme, which I'll talk about in a little bit. So this paper came out in Nature a few years ago. It was the first time someone had tried to combine essentially synthetic hydrogels with these organoids. So as I mentioned, the standard way in which you culture all these organoids since they had been discovered was in Matrigel. And I told you that Matrigel is derived from a mouse tumor. So because it's derived from a mouse tumor, it's a biologically derived product. It has massive batch-to-batch variability. There's some reports that the variability in the proteins that comprise Matrigel can vary up to 50% between different batches. And also it has loads of undefined components. And so if you want to do controlled experiments, it's very difficult to do that using a matrix that's very poorly defined. And so what they did in this paper is they started to look at, if we use synthetic hydrogel instead of Matrigel, what are the properties we need to have in this in order to culture these intestinal stem cells and then have them form organoids so we can do more controlled experiments. And this one here, what they're doing is they're taking essentially these LGR5 positive stem cells, putting them in these synthetic hydrogels, and they found that you need actually a stiffer hydrogel to allow the intestinal stem cells to expand. But then interestingly, in order to get them to bud and form the crypts that you then have these stem cells at the base of, you actually need to have them not in a stiff hydrogel, but essentially in a degradable hydrogel. So a hydrogel that softens over time. And so here they've put them in essentially these 50% or 75% degradable hydrogels, and then they form these really nice crypts. And they were able to show that they could do this. And this was all being driven again by these hydrogel cues. And so they summed it up really nicely. To expand the stem cells from a single cell, you need a stiff matrix that's non-degradable, but to form the organoids, you need a soft matrix that essentially degrades. So this sort of takes us to, now we can do things like experiments in synthetic hydrogels like that made from PEG. And I want to take you back to what I was talking about with these bidirectional interactions between cells and their secreted matrix. So we had done that before, and we knew this secreted matrix could influence cells in addition to the hydrogel cues we were giving them. And so we started to think, there's this work with intestinal organoids. We know that cells secrete matrix, and it impacts them when you put them in these 3D systems. So we started to think how we could try to use these concepts in disease modeling. And the obvious thing is to look at diseases in which you have pathological matrix remodeling, things like fibrosis. Sorry. The disease we started to look at was Crohn's disease. So Crohn's is a form of inflammatory bowel disease. It's marked by having chronic inflammation, but it's the chronic, and that has people have diarrhea and abdominal pain, long-term issues with malnutrition, lots of problems. But what really gets these people is not things like not directly the inflammation that it's causing pain and such. It's that long-term, many of these patients go on to develop pathological matrix remodeling around their gut, most often strictures, and these strictures require essentially emergency surgery to have sections of your gut removed because you end up with an intestinal blockage. And so we really wanted to focus to see if we could use these models to focus on this fibrosis in Crohn's disease. And so before I tell you exactly what we did, I also want to tell you that there's a specific type of immune cell called an innate lymphoid cell. There's actually multiple types of innate lymphoid cells, but these are tissue-resident cells. They're antigen non-specific, and they essentially reside at mucosal barriers. for example, in the gut, but they're also in the lung and other tissues as well. And the reason why we were interested in them is because it's known that if you have a healthy gut, the majority of the ILC in your gut are ILC3. ILC3 have been implicated in the past in supporting epithelial regeneration. But if you have Crohn's disease, a large fraction of the ILC in your gut are actually ILC1. But it's not clear if this is just sort of a consequence of having the disease or if ILC1 might participate somehow in these pathologies associated with Crohn's disease. And so we included these in the model. So I'll come back to them in a minute. So what we started to do, and I won't show you too much about the mouse, but we started to make organoid immune cell cultures. So you'll remember when I told you you could make pluripotent stem cell derived organoids. These have epithelial cells, but also mesenchymal fibroblasts. And so we started doing co-cultures of these sorts of organoids with ILC1, these immune cells that accumulate in the gut of patients with Crohn's disease. And we could use our synthetic hydrogels to start to understand the role of ILC1 in sort of these Crohn's-like models. But as I mentioned already, this had always previously been done in Matrigel, which is poorly defined. The manufacturer adds unknown concentrations of MMP inhibitors. Very difficult to study things like matrix remodeling in an undefined matrix. So we looked to create synthetic matrices to do this. So I won't spend too long on this, but basically the standard design for making a synthetic hydrogel is to do this. You take a peptide, which is normally degradable by MMPs, and it has thiol terminating peptides on each end, and then you mix this with a functional PEG, and then it forms a hydrogel. But if you want to make a soft hydrogel that's soft like the native intestine, you need to have a low polymer concentration. And at a low polymer concentration, if you use this design, the favored reaction in the hydrogel is for the first end of the peptide to react with one arm of a PEG molecule. But then because the next PEG molecule is far away, it will often react with a second arm on the same PEG, forming what's called a primary loop. And this does not help you form a hydrogel. And so what happens is it's very difficult to form a soft hydrogel that's soft like the intestine. So what we did is we worked with Chris Lorenz, who's in physics at King's. He helped us develop an alternative chemistry for forming the hydrogel based on using heterobifunctional peptides and a two-step reaction. And he was able to show really nicely using his molecular dynamics models that our new design allowed for the formation of many more network forming crosslinks than the standard designs because we avoided these primary loops. So at least 25% of the crosslinks formed in the standard design essentially don't contribute to the network. They're these primary loops, which we don't get in this system. So it's really nice work. We were able to make these hydrogels. We can make them very soft in the range of hundreds of pascals if we need to, which that's probably even softer than the native intestine, but we can model lots of tissues, being able to make them this soft. We also developed techniques using the atomic force microscope, using thin hydrogels with the organoids inside and probing the surface to understand how cells were modulating the matrix around the organoids. Because remember, we have fibroblasts surrounding the epithelium that are altering this matrix around them. And we also developed techniques using something called multiple particle tracking microreology, which is the idea, sorry, it uses basically this idea that a very small bead in a liquid will move by Brownian motion, whereas if it's in a solid, it will not move. So then based on that, you can use the Stoke-Einstein relation to relate bead movement to essentially whether you relate bead movement to sorry, it's hard to explain. You come up with this alpha value, which will be an indicator of which is a quantification of bead movement, which is an indicator then of whether the gel surrounding the particle is intact, essentially behaving as a solid, or if it's been degraded and is essentially behaving as a fluid. So if you have a cell embedded in a gel, essentially the beads move very, very little. As soon as the gel is degraded, the beads move a great deal. Your alpha value will go from essentially 0 to 1, and you can quantify that. So what it would give you around the hydrogels, there it is essentially this in situ mapping of where degradation is taking place. So you put your beads around your organoid, you have your fibroblasts around here, you track bead movement, and you can essentially identify areas of degradation and matrix remodeling. This is what it looks like in practice. Here you have an intact hydrogel, essentially the beads don't move. When it's degraded, they essentially move by Brownian motion, and you do this in the presence of the cells in the organoids. So when we put this all together, what did we find? We could use the atomic force microscope. We were seeing that this is a measure, this here is Young's modulus in pascals. Here's organoids only, organoids with ILC1. What we found first is that the median elastic modulus of organoids with or without ILC were essentially the same, but we found that in the cultures with the ILC, which we know accumulate in patients with Crohn's disease, you essentially had many measurements that were significantly stiffer, which we could see in patchy areas around. Here's the organoid's epithelium. We could see that we had upregulation of fibronectin expression, and on our immunos, massive expansion of the area where the mesenchyme was. And then we could see the opposite. If we used the multiple particle tracking microreology, we had lots of these measurements were very much softer than the median, and we knew this was driven because we could see it in the microreology by an upregulation, or I guess a significant increase in this alpha value, which is an indicator of bead movement and degradation. And we knew that ILC1 from single cell sequencing data sets secrete MMP9. I show you this story because basically using these ideas of understanding mechanotransduction and building 3D models and using synthetic hydrogels to do this, we're actually able to identify something using human cells and human disease models that you could have never found using a mouse. And that was, it looked like the ILC themselves were driving extracellular matrix remodeling around these organoids. And the reason why that was important for Crohn's disease is because we know these ILC are in the tissue. They're probably there as some sort of wound healing response, but our data was suggesting to us that when you have large numbers of them in the gut, as you get in Crohn's disease, it could be that their normal wound healing response could be tipping and driving something like the fibrosis that you see in these patients. And so it was really exciting that we were able to build this from these in vitro models and all of this understanding over many years about how physical cues and using materials to mimic a 3D environment for cells and now into organoids really allowed us to discover this. So I'm just about at my hour. I will finish by just showing some people from my group because I showed some data from my own group. Somehow the person who did all of this organoid work, Geraldine, never made it into a group photo, so she's just on her own on the side. And a little bit of the work I showed you as well came from my former postdoc, Sylvia Ferreira, who's now at Imperial. So I'll just stop there. I'll try to stop sharing so I can see you again, if I can. And I'll come back in and I can take any questions you might have.
Hi. Oh, hello. Thank you. Very nice talk. Seminar. So I wanted to ask about, because it's a very nice model, but I was wondering for the guts, very important is also like the muscle layer below and like all the peristalsis. And I was wondering if this is possible at any point to implement as well, because this like adds up the mechanics as well.
Yes, yes. And I get similar questions to this from people in lots of fields, because if you're a person who studies neuroimmune interactions or neuronal interactions with the gut, you also come and tell me like, but the most important thing is the neural innervation, or you come and tell me, well, the most important thing is the microbiome, or the most important thing is something else. and the answer is all those things are very, very true. And the model we've made here with a focus on the mesenchyme focuses on the mesenchyme, and it focuses on one type of immune cell. And if you talk to immunologists, they say, what about macrophages? What about every other type of immune cell that we know plays a role here? And so the answer is, we have a reductionist model. We take small steps in building these things. And there's a tremendous amount of research going on right now to make models more and more advanced. And these will include things like, there's papers that have come out over the past sort of year and a half where they've sort of opened up organoids and placed them along tubes, essentially, to be able to have movement through them, so that you don't have these organoids that essentially fill with the apoptotic cells from the top of the villi structures, and then they explode or die and you have to passage them. And that's not like the native intestine. So those are out there, and people are trying to do exactly this: put immune cells on one side versus the other, put microbiome in, try to include neuronal cells, try to include something like a smooth muscle layer or blood vessels and things. And the answer is, every time someone takes one more step like that, you do get a Nature paper. And there's, you know, champagne and chocolates on silver trays and things, but it's only still a small step, and it's nowhere near as complex as the native intestine. I think for us, we were lucky. We found one immune cell, the mesenchymal compartment. These interactions allowed us to identify something really cool, but there's so many steps to go before we can actually model the human intestine and ask every question we want to ask.
Okay, yeah, thank you. Yeah, it's cool.